The action potential. Again, show your hands if you've touched the action potential in an earlier class. Okay, I promise you I'm going to go into much more detail. Okay, and then I'm going to show you how to apply this stuff. All right, action potential. An action potential is that nerve impulse that I kind of explained to you about when I talked about putting a electrical wire into the outlet that's connected to a lamp. When you plug it into the lamp, it sends that electrical jolt down the wire and turns on the light. That electrical impulse is what we call an action potential. It's just one jolt of electrical impulse. It's like taking the plug, putting it in, taking it right out. One jolt going over there. If one jolt or one action potential, one electrical jolt, it's just not going to do enough work. It'll just maybe flicker that light and then it goes right off. You need a brigade of these electrical impulses going down that wire. So you're going to need a brigade of action potentials going down here to actually do some kind of energy thing, whatever it's going to do, contract a muscle, squeeze a gland, whatever it's going to do. Okay, so we have this nerve impulse that travels down from the dendrite, goes into the cell body, and then it goes down the axon until, when we're talking about skeletal muscle, so it eventually will go right to a muscle. That portion of the muscle that axon terminal touches, and I'm going to put touches in quotes because it doesn't actually touch, but it gets very close there. That portion of the muscle is called the motor end plate. Okay? And when it does, it's going to then spread the action potential across the whole sarcolemma, the cell membrane of the muscle. And I kind of showed you that in earlier slides of this presentation. We got to talk about the action potential down this axon too. Once it reaches the axon terminal, the end of the axon, like I said, it gets very, very close to the motor end plate, but it doesn't touch it. There's a gap there. And at the end of the axon terminal, it's going to send a chemical across that gap to the motor end plate. That chemical is called a neurotransmitter. Neurotransmitters, there's probably about, my guess is about 50 or 60 of them throughout your entire body. We're only going to be dealing with about a dozen of them. But the one we're going to be dealing with right now, when we deal with this neuromuscular junction, this neurotransmitter is called acetylcholine. All right, that's that chemical that comes off the nerve to go to receptor on the muscle. Okay? Now, we do have cells that are electrically excitable. Neurons and muscle cells are these good examples of it. They have a voltage that changes due to the stimulation. So this voltage is really a difference between one point and another point. The 2 points we're talking about is inside the cell and outside the cell. For instance, if you have let's say, I'm just giving an example, let's say positive 30 inside the cell, and then you have negative 70 outside the cell, then the charge, the voltage, is going to be different on two ends. The difference is going to be that negative 40, between negative 70 and positive 40. Or positive, did I do the math right? You get the picture though. It's the difference between the two. Okay? And the membrane potential is that it wants to go to zero. It has the potential to become zero. Okay? If it's not zero, it's gonna want to go zero. And that's what's actually causing this electricity to go on. If it's zero, it's not going to do anything. Like distilled water, it has no positive or negative charges in there. It's zero. It's neutral. So you can't really conduct electricity in distilled water. But if there's some charges in there, salts, it will conduct electricity. The resting membrane potential, because it's not zero, is going to be between, that difference that we just talked about, the difference is going to be somewhere between negative 90 and negative 60 millivolts. It depends on the cell that we're talking about. But our magic number that we're going to use is negative 70. This is the resting membrane potential for nerve and muscle cells. So at rest, this means that it's going to be negative 70 millivolts across that membrane. Meaning that inside the cell is 70 millivolts less than, or less positive than outside the cell. Okay? You're not going to have positive 5 outside the cell and negative 5 inside the cell, because that'll be 0. So it's going to be more negative inside the cell. How much negative? Negative 70 millivolts. Okay? Now, an unstimulated cell. It's going to be polarized, charged. And we're going to see a difference between outside the cell and inside the cell at rest. Okay? The best way you could understand this is using the salty banana that we've been talking about. Okay? So, let me show you what's going on here. Alright, again, salty banana, we're just going to do this really quickly, if you could remember this. Alright, it's a very happy banana. I make this fun for you guys. Inside the banana, what's the major cation? Potassium, right? Then bananas are loaded with potassium. So we're going to find more potassium inside the cell than outside. There's going to be potassium on the outside, but not as much as we do on the inside. Now, we do have potassium gates that are going to be over here. And they're closed. If the potassium gates open, which way does the potassium want to go? Outside. Do we agree with that? And that's simple diffusion. More potassium inside than outside, it goes from a high concentration to a low. If the potassium leaves the cell, does that make the cell more negative or more positive? Potassiums have what kind of charge? Positive. If the positives are leaving, that's going to make the cell inside more positive or more negative? More negative. Does that make sense? Okay. The sodium chloride from the salt. That means we have more sodium out here. And more chloride. I'm just going to worry about the sodium right now. And there is some sodium in the inside. Not a whole lot, but there is. also have sodium channels or gates. And these are closed. If the sodium gates open, which way does the sodium want to go? Into the cell or out of the cell? Into the cell. There's more sodium outside, so it's going to go from a high concentration to a low concentration, and it's going to go into the cell. If sodium goes into the cell, does that make the cell more positive or more negative? Positive. Is that clear? Okay, I'm going slow for you. This is where students start losing it because they deal with chemistry. A lot of students don't like chemistry. But I'm also going to throw a graph up here, and that really throws people off because students just don't like graphs. But you're going to love this one. Okay? Do we understand this concept? Why it becomes more positive? Why it gets more negative? Okay. So we have a lot more sodium on the outside, and inside we have a lot of potassium. Now, remember I said that the charge inside the cell is going to be negative 70 millivolts. It's a negative charge. Well, we have a lot of anions inside the cell more than we do outside the cell. Certain proteins, nucleic acid, phosphates, these are negative charges. And these stay in the cell. So because we have a lot of negative charges that stay in the cell, that's going to contribute to why inside the cell is more negative than outside the cell. They can't leave. There's another problem that we have. These channels that I drew over here, even when they're closed, they're leaky. Meaning, if you take your faucet and you turn it all the way really tight, it's still going to go drip, 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 drip. So even though we have sodium-potassium channels that are closed, they're leaky. They're not perfect. And some sodium is going to leak into the cell. Does that make the cell more positive or more negative? Positive. And we're going to have some potassium leaking out of the cell. Does that make the cell more positive or more negative? Negative. However, we have a lot more potassium leaky channels than sodium channels. For instance, I'm just giving you an example. If we have one sodium that's leaking in, we have three potassium leaking out. Net-wise, what makes that inside? More negative, more positive? If we have more potassium leaking than what we have sodium coming in, is that going to make inside the cell more negative, more positive? Negative. So because of this, this is also going to contribute to inside the cell being more negative. Okay? So sodium wants to leak into the cell, potassium wants to leak out of the cell. And that's just simple diffusion. So we have more potassium wanting to leak out of the cell than sodium coming in. That's going to contribute, all this is going to contribute to a negative 70 millivolts or negative charge as a resting potential. Okay? Questions so far? All right. So potassium wants to go out, sodium wants to go in. Now, what happens when a cell is activated? Okay? So, we're going to do our graph. This is charge. And we call this millivolts. This is time over seconds. And we have negative 70 millivolts here. We're going to have 0 millivolts over here. Does that make sense? Okay? Now, here's the resting potential at negative 70. What happens here is that sodium is going to start coming in here. And once it reaches what we call a threshold at negative 55, leaky channels are allowing sodium to come in. But once it reaches negative 55, because positive's coming in, this is going towards 0 now, right? Once it reaches negative 55, the sodium channels open all the way. Sodium is going to pour inside, make it inside the cell more positive. Now your book also likes to use other terms, likes to say, well, it's becoming less negative. What the? It doesn't matter. More positive, less negative. Just be careful, because when you in different books, lab books, textbooks, your teachers in A&P too, they might use different terms. They mean the same thing. More positive, less negative. And the sodium channels are opening wide, and sodium's going to pour in, making it more positive, and it keeps on going up until it actually goes past 0 to about a level of positive 35. I've seen positive 30, positive 40. We'll just go right in the middle. It reaches positive 35. Once it reaches that, once it goes past negative 55. We call this depolarization. And it's due to sodium channels opening all the way, making inside the cell more positive. Then at the top here, sodium channels close very fast. They open very fast at negative 55. They close very fast at positive 30, 35. And at this point at positive 35, as sodium channels close fast, potassium channels open fast. If potassium channels open fast, which way is the potassium want to go? Out of the cell or into the cell? Out. Does that make the cell inside more negative or more positive? Negative. So when that happens, it's going to go straight down. We call this repolarization. And that's due to potassium leaving. Okay? It's making the charge inside more negative, or other people like to say less positive. Same thing. Okay? Now, interesting thing that happens here. When the potassium channels, when they close slow. Again, at negative 55, where depolarization is going to really start, sodium channels open fast, they close fast. At the point here, potassium channels open fast, but when you get down here, they close slow. This is key. When they close slow, if this is a door that's over here, okay, if there's a door here, and there's 50 students inside the room, and there's like 2 students outside the room, and there's a door here, all right, think of a door like this. If I close, and students want to leave the room, because it's going to go from a high concentration to a low. If I do it very fast, I'm blocking all the students from coming out now. But if I close the door slowly, ain't I getting a few students squeezing on out of here? A few more? 3 or 4 students? So what happens here is because it closes slowly, you're still going to sneak out a few more potassium ions, even beyond -70. So when it closes, it's actually going to go even further underneath. It undershoots is what we say, to about -85. We call this hyperpolarization. Some people call it after potential. At this point, at -85, the potassium channels are now completely closed. They snuck out a few more potassium ions between -70 and -85. Now it's closed at -85, and now we got the leaky channels of sodium potassium going on again. And that's going to get it right back up to our resting potential of -70. Okay. So here's the parts again. -70 is a resting potential. They're leaky. That's what's causing us to stay at -70, amongst other things. But then the sodium channels are going to start opening a little bit until it reaches -55. Sodium starts pouring in a little bit. Once it reaches -55, the sodium channels open very fast and allowing a lot of sodium to come in, depolarizing the cell, making it more positive. Until it closes, the sodium channels close, and that's about +30, +35. At the same time, potassium channels are going to open very fast. And you will have tons of potassium ions leaving the cell, making the cell more negative. But these channels, the potassium channels, are going to close slowly, and because of that, it's going to undershoot. It's still going to go below -70. It's about -85. We call this hyperpolarization. Right? This is repolarization. Here's hyperpolarization. Until the doors of the potassium channels close. That happens at about -85, and now that starts balancing itself with the leaky channels, and then you're going to slowly get back up to -70. So an ash potential is a quick up and then down. All right, that's one cycle. Okay. The resting membrane potential is like this. It's stable voltage, and it's just waiting for the muscle or neuron to get stimulated, to do something. It's like that little tickle in your nose. It's just getting ready to sneeze. Okay. And once one action potential takes place, it kind of tells another one to do the same thing. It's a domino effect. You can't just have one action potential. It's not going to do any work. But if you have a brigade of them happening, like dominoes, a cascade of events, boom, 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 then that's what will happen. So positive goes in, positive goes out, making this graph go up and then back down. That's one action potential. Won't do any work. You got to have a bunch of them, a frequent amount of those, one after the other. They can get a little bit complicated. I was trying to show you with gates over here. There's activation gates, inactivation gates. You'll see that with a little video explaining that a little bit more. Like I said, it's a cascade or a domino effect. And it only goes one way. It doesn't go the other way. We'll explain that too. So here you have one action potential going here. Sodium goes in, potassium goes out. It goes along the action potential. So it just went a distance. And there's going to be one right behind it. So there's the first impulse. Here's the second one. And behind all this, we're going to have the ATPase pump, the sodium-potassium pump, which we're going to explain that too, the purpose of that. Okay? So just showing you on here, positive goes in, potassium goes out, and it goes along here. Okay? Any questions so far? All right, now let's talk about the threshold. The threshold is that as sodium comes in, it has to reach this negative 55. If it only makes it to negative 60, we're not going to have an action potential. The action potential will occur once we reach negative 55. Okay? If it doesn't, then we say it's sub-threshold. And once you have an action potential, you have the action potential. In other words, once we reach negative 55, you will have the action potential. You're not going to get a partial action potential. You either fire an action potential or you don't. Okay? We call this the all or none principle. It doesn't go higher than positive 35. It doesn't go lower than negative 70 for an action potential. I mean, here negative 85, yes. But I'm saying you can't make a stronger action potential. That doesn't happen. You can't make a weaker one. You either have it or you don't. An action potential is an action potential is an action potential. They're all the same. It's just you could have a more frequent action potentials occurring or less frequent. Okay? So there's no such thing as partial action potentials. Alright? And this just explains to you about sub-threshold. If it doesn't reach negative 55, you will not have an action potential. So we're going to talk about this little area here, which is called the local potential. We'll talk about that later, but we'll talk about that as it go up and down there. Okay? Now, let's talk about refractory periods. And this will explain why the action potential only goes one way. It doesn't go backwards. The best way I can explain what a refractory period is, well, we have two types. We have an absolute refractory period and a relative. And the way I explain what this is, not many teachers do it this way, is think of your toilet bowl at home. Okay? Your toilet bowl, I said it right. Not the toilet bowl that's in the school here, the college here, because that's a turbo flush. Okay? I'm talking about the one where you just hit the handle and you gotta wait to fill up. Now, when you flush the toilet at home, when you flush the toilet, you bring down the handle, and you can't flush it again seconds later. Does that make sense? You know what I'm talking, and don't say you don't use the bathroom, everyone here uses the bathroom. Okay? But what happens here, you flush the toilet, now you can't do another flush until the tank fills up with water again. It's in an absolute refractory period. Does that make sense? So there is an absolute refractory period here where you can't fire another action potential until that action potential that we just did leaves its refractory period. You can't flush that toilet until the tank fills up. So we call that an absolute refractory period. All right, sodium gates cannot reopen until the cell repolarizes. It has to go back down to here. Meaning that if I do a stimulation that occurs over here, I'm not going to have an action potential that happens again and going up like this. Not going to happen. It has to reach negative 70 again before you can hit another action potential. So that's why we can't do another one if the other one is behind it is going into a is still in the refractory period. Yeah.
Is that the negative 55 or negative 70?
Negative 70. If I do this at, let's say, I don't know, negative 40, it's not going to do anything. It still has to reach negative, it has to reach this refractory period, or the end of the refractory period has to reach here. So if I stimulate this cell over here, I can't, it's not going to do anything because it has to reach negative 70 again for it to happen, for another action potential to happen. That's why we can't, the action potential only goes forward. The one right before is still in a refractory period. Okay? Does that make sense? All right? Now that's a absolute refractory period. But then we also have something called, I just explained all that, a relative refractory period. This is where, think about it, again, we'll talk back to the toilet bowl. You flush the toilet, you know, it goes all the way around, and then the tank is filling up. But you know how, like, you hear the tank filling up, and it's just about almost done filling up, but you can flush it again. You do a nice hard flush, it'll work again, because it filled up a certain period. Maybe 3/4 of the tank has to be filled before you can actually flush it again. Right? You all know what I'm talking about? Okay? So that's what we call a relative refractory period. A relative refractory period is that we can actually do, if it's strong enough, if it already reached negative 70, if we stimulate the cell, here at, let's say, negative 80, if it's strong enough, you're going to have to get it now to still negative 55, and then you'll do it again. So instead of going from negative 70 to negative 55, which is 15 points, right? Am I doing the math right? 15 points. If it's at negative 80, now we got to do 20 points to do that. Am I doing that right? No, 25 points. Does that make sense? So you're going to have to do, you could actually stimulate this cell again and find another action potential, but it's got to be a stronger stimulus than what it was at negative 70, because now you're at negative 80. So you got to kick it up even further, because it's that much farther away from negative 55. And we call that a relative refractory period. And it happens during hyperpolarization. So this here is showing you where the absolute and relative refractory periods are. The beige area here is the absolute refractory period. There's no way you can actually fire another action potential during this phase, or during this duration. But you could have a relative refractory period during hyperpolarization, but you're going to have to put a very strong stimulus, because now you're going to have to go, instead of negative 70, negative 55, you're going to go like negative 80 to negative 55. But it can be done. Does that make sense? Did the total ball help you? Okay? Questions on this? All right, now, we got to restore the resting potential. See, sodium went into the cell. Potassium went out. If that keeps on going, then there's going to be it's going to be isotonic. We're going to have 50% of potassium outside and 50% inside. When that happens, the cells are going to do any kind of action potential. Does that make sense? Because it's 50-50. Which way is the sodium and potassium want to go? No place. Because it's equal on both sides of the membrane. So we got to somehow put the potassium back out of the cell, and somehow we got to put potassium back into the cell. But we're going against the current for that, or against the concentration. So we got to restore the electrical chemical balance. So we got to somehow make, because the cell is positive inside, now that sodium poured in, we got to correct that. And we just saw how that's corrected by making the potassium channel open. The potassium goes out. All right, so that's what happens. Repolarization will fix that. All right, and that happens with diffusion. But to get the ionic balance, in other words, to get the potassium back into the cell, to get the sodium back out of the cell, because like I said, this is the way it was at rest, right? We have more sodium out than there is in. We have more potassium in than there is out. But when these gates open, potassium is going to go out until it closes. And if that keeps on going, it's going to eventually get 50-50. And then the potassium doesn't want to go out anymore. Because you're going to have 5 inside and 5 outside. You need, 9 inside and 1 outside. Right? That's what diffusion's all about. Once it's 50-50, it's isotonic, it's not going to go anywhere, which means you're not going to have an action potential. So we got to find a way to put the potassium back inside. We got to find a way to put the sodium back outside. But the sodium is going to go from not natural, that's not diffusion. The potassium wants to go from a low concentration to a high. So in order to correct this, we got to do a different form of a transport mechanisms, because diffusion is not going to work like this. We're going to have to use ATP, active transport, to do this. Okay? So that's what I just explained over here. There's a problem. We have to correct this. Okay? So we got to do this. And the way we're going to do this, who here knows how we're going to do this? What's the pump we're going to use? ATPase pump, the sodium-potassium pump. Okay? The sodium-potassium pump is going to correct this. It's going to put 3 sodium outside the cell and 2 potassium back inside the cell. Right? Na-na-na, out. Na-na-na, out. KK goes inside. Right? That's what's going on here. Why is it 3 and 2? Why isn't it 3 and 3? Why isn't it that 3 sodiums going out and 3 potassiums going back in? I mean, that's going to put the sodium and potassium where they're supposed to do, sure. But why is it not an equal amount on either side? 2 out, 2 in, or 3 out, 3 in? Why? Excuse me? The inside needs to be negative. So if we do 3 and 3, if 3 positive goes out and 3 positive come in, what's that going to do to the charge? Nothing. Right? Does that make sense? It's not going to do anything. You're putting the ions back where they're supposed to, but you're not doing anything to the charge. So we got to have more positive going out, as Fema said, right? Okay? Feba. All right? You want more potassium going out, making it negative, and I'm sorry. You want more sodium going out, making it more negative inside, and a couple of potassium going in. And that'll still make it negative. All right? Does that make sense? All right? So going against the gradient, you need active transport. ATP is used for this. And this is always working behind the action potentials to put it back. Otherwise, you're not going to have an action potential. Okay? So again, why is it not 3 sodium out and 3 potassium in? It's because you need that charge difference. If you do that, it's just going to be equal. And that's not what we want. Okay? So we need to have more positive leave the cell than we do entering the cell to keep it negative. Okay? And this is just showing the sodium-potassium pump, which you've seen before. Okay? Again, 3, sodium out to potassium in. Okay? And you saw this before. Here's the action potential going down the axon. Here's the second one. And then there's going to be a sodium-potassium pump happening in each one of these action potentials behind it.
We have looked at the resting potential in a neuron that is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus changes the permeability of the cell membrane to sodium and potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal, called an action potential, that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close depending on voltage changes across the membrane. When no nerve signals are being transmitted, these channels are closed. A stimulus causes voltage-gated sodium channels to open, and sodium ions rush into the cell. The cell becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The cell returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. In the wake of the action potential, potassium leaves the cell, restoring the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting potential distribution of sodium and potassium ions. Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.